right. Hello, everyone. Welcome on in to another episode of GSA on Twitch. My name is Belent, and today we have Professor Nele Haelterman with us, and she will be chatting to us about her career journey to up to this point, which actually involves being part of the early career leadership program at the GSA, a mentor there, a science writer, a professor, researcher extraordinaire. Um, she's got a
all over the place.
no, it's a pretty inspiring story, and we're looking forward to talk with you about it. So I think we're just going to go ahead and start off. If you can tell us a little bit about where you are right now, and then we'll go back in time of how you got to this point.
Sure. I'm based in Houston, and I am an assistant professor here at Baylor College of Medicine. I am part of the Rejoin Consortium, so we study joint diseases like knee arthritis, and mostly we try to understand why people feel pain. So we're trying to figure out what happens within the knee and how innervation changes, so how the neurons change within the different tissues in the knee and how that causes pain. Like you mentioned, Belinda, have been a little bit all over the map when we talk career-wise. I've been in this position now for about a year. And before that, I was working for The Scientist as editor, as an assistant editor there. So I was a science writer, and we were in charge of developing a bunch of different science communication projects. So think about any kind of way that you could communicate a message, and we were doing it, except for Twitch. We weren't doing those. But we had blogs, we had articles, podcasts, we had posters, and a bunch more things. So that was a great experience. And then before that, I was a postdoc in human genetics, looking at skeletal development and skeletal diseases like osteogenesis imperfecta, which people know better as brittle bone disease. And I was studying that. I had mouse models for that disorder that we were studying. Before that, I was doing my PhD, and that was in fruit flies. And there I was studying muscle development. So I have my PhD in developmental biology, and I was studying innervation and how motor neurons develop, and then how the muscle develops along with it. So yeah, all over the map. I tend to follow my interest in that sense.
There,
I haven't stuck to a single field or organism.
but it does seem like there is a link between them, right? Like there's skeletal structure that you're going into, but also like muscle building on the skeleton from the fruit flies. There's a theme. So how did you, I guess, originally get interested in a scientific career, and what made you kind of settle on this trajectory of almost ending up with human disease models, or was it not settling to begin with? It just over
Yeah,
time.
it's a good question. I've thought about it a lot in the last few years. It's mostly been opportunities that have presented. I've definitely had my own interests that I've pursued, but I've always stayed open to changing fields depending on what opportunities I had or what I thought might. Maybe it's weird to say, but might have a better chance of me continuing in that path down the road. So I initially, this is way back, when I had to decide what to study. And so I'm originally from Belgium, and in high school I mostly studied languages. So I didn't have much of a scientific background. And so when I got to my last year of high school and had to decide what to start studying, I was really interested in law school. And I took one class. So we had, at the universities, you could sort of go sit in lectures to see what it would be like. And I went to one, and that was enough for me to know that that was not my path. But it sent me a little bit in a spiral of, okay, so if that's not it, I thought I had my road mapped out. So if that's not it, then what do I want to do? And I wanted to study medicine, and become a doctor. but I really didn't have the background for it. And so I really that summer studied very hard to. In Belgium, you have to pass this entry exam, and so I studied really hard, and I tried to uh, learn whatever science I hadn't learned in the 6 years before that on my own. But to give you an idea, I had not heard of, well, I had heard of what a pH was, but I didn't know how to calculate it or anything. So
Is
it was difficult.
is the school system in Belgium where you have like the high school equivalent, and then instead of college, you go straight to like medical school or any
Yeah,
other?
you go
Okay.
straight into yeah. So I tried and I failed that entry exam, and the equivalent, which in Belgium you could at that time, I don't know if that's still the case, but at that time um, you could start in your first year of biomedical sciences. The classes were almost entirely overlapping with those of the first year of medical school, and so you could take that entry exam again at the end of that first year, and then go directly into the second year of medical school if you passed. So you can already guess it. I failed that exam, and I started biomedical sciences, and then there was this whole new world that sort of opened up for me because I hadn't heard much about um, the the research world and what it would mean to be a scientist. But then I got exposed to it there, and I actually really liked it, and so I just stuck to it.
So
And then yeah, to get back, I guess, to your question of like how does that tie into it? I was very interested in developmental biology. That was always a class that I liked the most. Um, and so I looked for places where I could do my PhD in developmental biology. And so then that interaction between nerves and muscles was really interesting to me. So I pursued that a bit more.
so at the end of that first year at that university, did you apply for the medical school track, or were you just full gung ho on the science
Um,
track?
no, I did. I still did. But then when I had to make the decision, I just liked science more, so I stuck to biomedical sciences in that second.
And so was there a developmental biology class? Like I guess during that
Yes, but
first year.
it was only so in Belgium the in all of Europe now the university system works with a bachelor's and a master's, kind of like what you can do here too. But in Belgium or in Europe, it's sort of implied if you start university, you'll do both. So you'll have 5 years there. And so developmental biology only came in my first year of my master's. And I had a great professor there, and a really cool lab where uh, we got to manipulate a chicken embryo as it was growing and see the effect of what would happen if you would remove the cells that give the growing signals off of the limb bud as it's developing. And so you could see the changes in that one limb bud there, and that was really powerful and was really um, yeah, powerful and convincing
That's
to try
really
and
cool. I
understand that.
What is it like working with chickens? Just as a naive question, because it's I know you usually hear
You
like
do most of the work in the eggs themselves. So there's I think right now there are not as many people studying chicks as there were before, but they are you can actually electroporate constructs during very early in development, and they are very accessible. So compared to a mouse embryo, which is in the uterus, you can't do that much to manipulate it. But a chicken, you cut like a small window in the eggshell, and then you can electroporate. There's been a lot of work in spinal cord development that's been done in the chick, and so you can very specifically manipulate this small section of the spinal cord, or wherever you're studying, and then you just close that back up. Put like a little piece of parafilm or plastic on top of it. You put it back in the incubator so it stays nice and warm and it keeps developing. So it's I mean, maybe if there's some non-scientists here, maybe it sounds a little bit cruel, but it's a very powerful system to be able to make small changes and see what effect they have on the developing tissue, without having to um, run, let's say, 20 experiments in parallel to have different time points. You can do it all in the same animal.
Can you also look into the embryo and see what's happening with that little window that you cut?
Yeah, well, yeah, I'm sure by now the field has developed a lot. This was almost 15 years ago. So um, I am pretty sure that by now, with all the advances that we've had in the microscopy world and field and technologies, that you can do much more powerful time lapse images of really looking at how the cells are moving, even if you've labeled them.
Nice. And so another one more naive question on that. With the chickens, did you have to have a chicken farm in the lab? Like where were you getting the eggs from to be able to do the
They are,
experiments?
and they are ordered from a specific company that is. So yeah, they will have a chicken farm there for sure.
Okay.
They have. I am sure they have a ton of regulations that they have to go by to try and reduce the variability experimentally. But that is really outside of my my realm of expertise. So I'm just um, yeah, hand waving here.
No, that's a really cool. So you like that you got to work with such an organism, like getting the master's
Yeah,
degree.
yeah, and I did a rotation when I had moved to Houston. I did a rotation in a lab that was also working with chickens, and so just to expand on that point, we would get eggs on, I think it was, let's say, Tuesday and Thursday, and those were the days that you would have to electroporate and then harvest your embryos at certain time points after, depending on when you wanted to look. Um,
That's really really
yeah,
cool.
it's it there, it's every model organism has its pros and cons, and for the chick, you could the lab where I was rotating in was combining it with mouse models, and so there were certain experiments that they could do in the chick to get a certain amount of information, and then try some validation experiments in the mouse. um, so yeah, those were really cool experiments.
That's a really cool transition between the two organisms and like a nice back and forth. And it seems like the modern day fields are going that direction too, of validating in different ways. So
Yeah.
you were working with chickens. You decided you wanted to get your
Yeah.
PhD, like furthering in developmental biology. So how did you decide on where to go? And
Yeah.
going abroad as well, it seems like it's a pretty big leap. And we've had people
Yes.
on the
Yeah.
screen before who've talked about like that transition from either Europe or Canada to the States. Like it's a big jump. Like how did you come to that
Mhm.
conclusion, and where did you pick?
So my two fields of interest were really genetics and developmental biology, and I did my so in Belgium, when you're in your second year, at least for biomedical sciences, when you're in your second year of your master's, you have to do a 1 year thesis project in a lab. So you also do these shorter rotations in your first year of your master's, and then you select one of those labs, very similar to how the PhD process works in the US. Um, and so then you choose one of those labs to do your master's project in. And the lab where I was working in for my master's thesis, I was working with fruit flies, and I was developing a fly model to study ALS, so one of the neurodegenerative diseases, and and that was sort of the second realization of, wow, this is such a powerful model organism. There's so much that you can do when it comes to the genetic tools that exist to study the function of a specific gene in the fruit fly. Um, and it's also really the model organism where a lot of the our current understanding of developmental biology has come from. So those two combined, I started to look. I wasn't at that point really sure whether I wanted to continue in the neurodegeneration field, or if I wanted to go more into the developmental biology direction. um, and I just started to look around at different people in the field and what people were doing, and I had selected several faculty that I might want to work with, and I was relatively naive, and I really didn't know much about how the system worked outside of Belgium. And so in Belgium, you really just email faculty and you tell them, I'm I'm graduating with my master's. I would like to do a PhD. Are you looking for somebody? And if they are, you can go talk to them and sort of start the process. And usually you'll end up, if they have funding, um, they can take you on for the first couple of months while you develop some preliminary data, and then you can apply for fellowship, um, and get going that way. And I did that to the faculty I was most interested in, who was Hugo Bellen here in Houston, and he's really one of the grandfathers of fruit fly genetics, and has developed so many amazing tools when it comes to fruit fly, to manipulation of genes and studying their function in the fruit fly. Um, and so I literally emailed him what I just told you. I'm X, Y, and Z. I've done this, and I'm interested in doing a PhD in your lab. Are you looking for someone? And he very briefly responded. He did respond, but he told me, well, we just selected our students. Too bad.
Oh, no.
Try again next year. And he did say that. So at that point, Hugo was the chair of the developmental biology program at Baylor College of Medicine, and so I in bits and pieces managed to piece together and learn that the system in the U.S. is very different, and that you have to apply to the university itself or to the college itself, and apply to the program or the department to get into grad school, and that then you rotate and then you select the lab, so you don't jump that whole process and go directly into the lab. So that was a bit of an eye-opener there. And I was very lucky to have a PI in my university, which was in Leuven. And he was a former graduate student of Hugo, and he had taught one of my classes. And so I went to talk to him, and I said, this is what I want to do. But he told me I'm too late. Now, I'm going to lose a whole year. What am I supposed to do? And so, that's he sort of guided me through it and told me, moving from Belgium to Houston is a pretty big move. They are quite different environments, so maybe you wanna see if you could work as a technician in his lab for a little bit, while you get used to the system and how it works in the US, and so you can also figure out if that's the right environment for you. So I emailed Hugo with that question, and long story short, at that point he didn't have money. I almost found another position where I could go, and then I still actually preferred to go to Hugo's lab. So my mentor in Leuven had told me, just send him an email and tell him, look, I have this opportunity. I have to decide relatively shortly, but I still would prefer to come to you. So has anything changed in your funding or in your finances? And he literally emailed me the next day saying you can start in November.
That's awesome.
And that was like a few weeks away, and then it was all the madness of trying to get that planned. But yeah, I think I got very lucky there with my mentor in Leuven, who managed to guide me through that, because I really didn't have much of a clue how things work differently outside of the environment that I was used to.
Yeah, and just how you're describing the European system as well might seem very strange to folks in the U.S. system, right? And so that
absolutely.
That cross difference it can be very difficult to navigate unless you have someone helping guide you. So that's a really cool story that it worked out, right? And it just persisted in those cold emails. Yeah. So we do have a question that'll be regarding how you were going through academia and science writing. But before we hit that question, let's jump into graduate school. So you said that you got to work with Hugo a little bit before the actual rotation process started.
Mm-hmm.
So what is your thought on that year that you were as a technician and doing the application process, and then how did that dovetail into the rotations, and how did you end up knowing where to choose? Like,
Yeah. Um, it was a fantastic year for me. I came in thinking I knew what I was talking about. I knew I had quite a bit of background in genetics. I had background in fruit fly, in working with fruit flies. I had some background in developmental biology, but it took just a few days, not even, it took one meeting with Hugo for me to figure out that that was really not the case, and there was a ton out there for me to learn. He put me on a project along with a really amazing scientist who at that point was a postdoc, and his name was Koen Venken, and he was really developing a lot of the new technologies for fruit flies. And so the one we were working on then was called Mimic, and it was a technology that would let you put this mobile genetic element that you would at that point. I'm not going to make it too complicated. It was a genetic element that you could use to either completely remove a gene function, and once you had it, so you could study it as a knockout and study what happened if you had lost that gene's function, and then you could convert it into just becoming a functional gene again, but now with a fluorescent tag, so you could see where it was expressed and where within the cell it was functioning. And then over time, there's a bunch more variations to that system that were developed, but it's become really powerful. So that was a really great project for me to work on as a technician, and it was. I think I would really recommend people interested in doing a PhD um, to try that out, to work as a technician in a lab first before applying. I know a lot of people come out of college and want to make sure they you I felt the same. It feels like losing time. You need to make that jump. You need to start. But I think that's really a misconception. I think it's really important that you try to figure out if this is for you before you do it. Um, and so I felt incredibly stimulated and super, convinced that this was the right track. So then I applied to grad school. I really did want to go to Hugo's lab, so I only applied to Baylor. And yeah, I got in. And then I did my rotations, and the same. I was super convinced. I only wanted to go to Hugo's lab, but I picked some rotations in different fields that I was interested in, um, so that I would have different experiences too. And then I found that actually um, I got very confused because I did like all of those rotations and I did like those environments. So I found it very hard to make the choice after those rotations.
do you by
What
chance
I
remember what fields they were in? Like, you have the fluid fly genetics with Hugo. Like, how drastically different were they?
They were all over. I was trying to have different model organisms.
Mm-hmm.
So one was, like I mentioned, combining the chick and mouse, and was studying glial development, glial cell development. The other one was combining fruit flies and xenopus, so the frog,
Okay.
to study early embryogenesis and how cell membranes form. And the last one was combining chicken and mouse also, but was studying the inner ear development. So was studying hearing, and they were all fascinating. So it was difficult to make that choice. But what I did, there was also a difference in the size of the lab. Um, and I did learn that I for myself thrived better in a bigger environment, like a larger lab, um, compared to a smaller one. So that was one of the things I think, when if you're at that moment trying to figure out where you should rotate, you can think about, don't just think about the PI. You should think about other factors too, like different model organisms, different fields. Some people come in to their PhDs with a very clear idea of what they want, but they also haven't experienced anything else. So I think it's good to give yourself. It's really that's what they are there for. So take advantage of that opportunity.
It's surprising, right, how something like a size of the lab could influence what your choice is at the end, because that isn't the PI necessarily, it's not the science necessarily, it's the environment. And I know when I was in school, it really wasn't talked about of what the environment should be and the importance of it, but rather it was just the science of it. And so it's nice to hear someone stressing that you should actually think about you too and set yourself up for being successful.
And it's hard, right? I think all of those elements play into it. It is also the PI and the science that is done. That's a large part of it still. Um, but the other factors which can be equally important contributors to your happiness in a lab environment, they're often not talked about. And I think those are really yeah, I think they should be brought up a little bit more so people think about it. Because once you join a lab, it's going to be for some time, and if you realize after that, I feel kind of in one of the labs I felt relatively lonely. It was a very large lab, and there were only 3 people there, and I could really tell that I didn't feel very motivated to do much. There was nobody else around, and it just felt, yeah, it was a pretty clear signal that that wasn't the environment for me. So yeah, I think that's important to consider in those choices. And I think the other thing it showed me was regardless of how convinced I had been of what I wanted before, I did really enjoy the other fields too, and I felt that if maybe I didn't have the background, but I could develop it. And that was something that carried through in all of those other transitions down the road. The fact that I think what a PhD does is really give you the skills to become an expert in a field of your interest. You will become an expert in the field of your PhD, but you also get the skills to learn things outside of that. And that's another thing that it took some time to realize, but I think we all have as scientists, that you learn how to find the tools you need to become more familiar with a skill or with a topic or a field. Yeah.
Yeah, I love that mindset of it. It's not just a terminal degree; it's something that helps you for the next stage and how you view the world, right? So why? So you chose Hugo's lab. It was the right size, right? It felt that it was maybe the right organism. What was your project like? What was the big thing that you're proud of in terms of the research out of the lab?
I worked on so many different projects in Hugo's lab. It we did this large screen where we would randomly introduce mutations in the fly genome, and we were looking for mutations that were um, that fell in essential genes, so genes that you need to survive. But once we had that collection of we had 2,000 different fly strains with mutations.
Mutations.
2,000, yeah, 2,000 different mutations, that after years of mapping and trying to figure out which gene they mapped to, um, ended up mapping to 500 different genes. So we had mutations in 500 different genes, and it was a collection that you could then sub-screen. So if you had a process that you were interested in, you could, as long as you had a good readout. For instance, one of the projects that I worked on was looking at neuromuscular junction development. So looking at how the neuron that innervates this one particular muscle in the larva, into one of the muscles of one of the segments there. It has a very characteristic pattern. And so we were looking at through those different lines to see which of these mutants have a change in that pattern. And so those were revealing different molecules that would influence neuronal development. And so, we had a lot of different screens that happened that way. I helped to develop the mapping technology. So at that point, now sequencing is of course much more um, of a default technology that people use, but at that point it wasn't yet. And so we developed a combinatorial technology to combine the classical mapping tools that people were using for identifying where mutations were lying in the fly genome, and combining it with whole genome sequencing so that you could really, instead of spending years mapping a gene, you could bring it down to in the best case scenario, I will say a couple of months. And so that was one of the large projects. There's a lot of different projects that stemmed out of there. But I ended up being interested in a mutation that had this really weird phenotype in the muscle, where in a fly muscle, a muscle is a syncytial cell, so it has multiple nuclei. Um, but basically, normally in a healthy fruit fly larva, um, you have those nuclei at equal distances from each other throughout. So if you think of the muscle, you stretch it out, it's a rectangle. So the nuclei would be really nicely separated throughout. And in this mutant, they were all like they had all fallen to the bottom of that rectangle, and they were all clustered together. And I thought that was so weird. I had never seen anything like that before. And I had looked at, hundreds and hundreds of larvae, and I'd never seen that.
And
Um,
could the
and
animal still move normally, like where its muscles
they were
functional?
slower, but they could still move. But
Okay.
this was a mutation that if the larva had lost or had that mutation in a homozygous state, so didn't have any healthy
Hmm.
copy of that allele of that gene, um, it would not make it to a grown-up fly. Some of them managed to eclose, so managed to crawl out of their pupal case. But if they did, then most often they would really, they would crawl out of the pupal case. They would fall on their side into the food, and then they wouldn't be able to get up, and then they would die. So their muscle function was clearly impaired.
Wow.
And so, long story short, it was an E3 ubiquitin ligase that was regulating the molecules that connect the nucleus to the cytoskeleton. And so, because that connection was impaired, the nuclei were not connected to the cytoskeleton, and were just, as the larva was moving and was contracting and expanding, all of those nuclei would just bundle up at the bottom of the muscle.
That's such a
So that's
weird
when I first got,
phenotype too. It's like I
yeah,
imagine you
yeah,
see it on the scope, you're like, what is this? Like, why are they all just
yes,
here?
it really took some time to figure that one out, and there it was the benefit of Hugo being so well connected in the fly community, and he had seen one other paper ever, before. um, and actually, it's maybe that's not entirely true. There, it was really like in around that time there were two papers that had looked at that concept of where because no one was even now, not that many people think of where a nucleus should be within a cell. You just, you know whether it's a bit more to the bottom of your cell or a little bit more to the top, you don't really think that matters much, as long as it's, pumping out the right products.
As long as it's there,
um,
right? We don't necessarily care
yeah,
where.
no, but it does matter.
Yeah.
So that was a cool project. It was a difficult project to work on, but it was really a fun project. Or yeah, fun and interesting. And we found that in humans, that was sort of the transition of Hugo's lab to try and connect um, the studying of gene function in the fruit fly to trying to understand what genes can be disease genes in humans, and then to try to connect the human genetics field to the fruit fly genetics field. And that has turned out, or has proven to be really powerful as well. So for in our case there, we actually found we connected with a scientist who, by chance, was had her lab across the street in the Texas Medical Center,
That's
and
very
she had
fortuitous. Yeah.
yeah, it was really hard to wrap my head around that one. But she was studying aortic aneurysms, and the familial kind of them. So she had several patients with mutations in the human homologue of the gene I was studying, um, and they had their aorta would rupture when they were kids. So this can happen, spontaneously when you're 50 or 60, but they would have it happen when they were 10 or 8 or 16. And it was really cool because when, I mean, that sounds bad because we're talking about patients here, but she had treated these patients and had managed to help them survive. Um, and as she had done surgery and collected a piece of tissue when she was repairing the aorta, we were able to do EM on them. So look really at the ultrastructure of this tissue with the electron microscope. And we had done that for our fly mutants too. And it was unbelievable because the shape of the nucleus was the same. It, instead of being nicely stretched out, it was really, you had these blobs and bulges everywhere. So that was yeah, really unbelievable.
That's amazing, right? When you see that uh, it's the same between those two organisms, and you're like, okay, I'm definitely on the right track here.
Yeah, yeah.
We have Nelf. Hello, welcome in. For those coming on in, we have Dr. Nele Haelterman here with us today, assistant professor at Baylor College of Medicine, and we're going through her career journey now. Currently, the graduate school years when she was making discoveries between conserved discoveries from the fruit fly to the human and back. And so, when looking at the nuclear structure in the humans, were y'all able to do any experiments to try to correct it in like a cell culture or
Yeah,
humanizing the
actually in the
fly?
fly. Yeah, so we humanized the fly, and we first introduced the human wild type gene and saw that it could rescue those phenotypes. And then if we instead used the mutation that the patients had, then we could bring it back. Then we could see that again we would have those same phenotypes in the muscle. So that really um, proved the point that those mutations were pathogenic and that they were causing the disease in those patients. Whether like it was a different muscle type, but the concept we thought was the same. That here these muscle cells were not able to sense the changes in blood pressure as much and weren't able to regulate the way we thought. Right, we didn't. Look into, or we weren't able at that time to look into it all that much further, but we thought that if you would have changes in blood pressure, that those are normally relayed through the cytoskeleton to your nucleus to change gene transcription, and that here, because the nuclei were no longer connected to the cytoskeleton, that wouldn't take place anymore.
That's
And yes, I see the undiagnosed disease network. Yes, that was at the birth of the UDN. um,
that's really cool because so for those who don't know in the chat, that's I think it's fair to say, right, one of Hugo's brainchilds of look.
yeah, yes. The Model Organism Screening Center, or in short, the MOSC that he developed as part of the undiagnosed disease network is really the um, the center that helps interpret the variants that are found in patients with a genetic disease, and that's really still one of the main challenges when you see a specific genetic trait that is uh, blanking on the word here, inherited from one family to the next, um, and you want to try and understand what has happened and what the pathogenic or what the causative mutation is. You can do whole genome sequencing, but you will still find a ton of changes between different individuals that could explain the phenotype or could explain the disease. So it is hard to figure out which one is really causing disease. And so with that's what the Model Organism Screening Center does, is it first goes and look at which variants do we have, what genes do they map to, and are those genes more conserved in whether it's the fruit fly or the zebrafish. Those are, I think, the two main organisms still of the mosque. So then they figure out, do we do the phenotypes themselves make more sense to study in the fly versus the fish, or is it the gene? Both organisms have their pros and cons, so they first figure out which one is better, and then they try to model it and humanize the organism to study that.
It's a really
It's a yeah.
yeah.
I was going to say
No,
the
it's just a
same.
really amazing technique, right? That you can go from, a broken fly essentially, right, genetically mutated. You put in the normal human copy, it fixes things, and then you put in the mutant human copy and reverts it back to how it was with the mutant fly
Yeah,
copy.
yeah. That's evolution right there for you.
Yeah. And it must
um,
have been really cool to be there at the start of that, because it's such a powerful tool, and also it's combating the big pharma, right? Because they're doing that for diseases that don't necessarily have a lot of money in it, right? But you just
yeah,
don't know what's wrong.
yeah, yes, and not many patients too. Sometimes they're like in our case for the familial aortic aneurysm that we were studying. We had 3 patients. By now, there have been more that have been found, but the UDN really tries to help patients who very clearly have um, a hereditary disease, and tries to they have a whole this is by now really a large network where you have a ton of clinicians and doctors who are involved, who are experts in all different types of medicine. So whether you have a neurological phenotype or a metabolic syndrome, these patients are very carefully phenotyped to really understand um, the full breadth of their symptoms, and then in parallel their genome is sequenced. I think by now, for depending on, they'll try to see um, what will help the patient most. But they can look at their transcriptome. They can by now do a bunch of things to try and figure out what is causing the disease in these patients, and what might actually help. Because the cool thing is, once you know the gene and the mutations that are causing disease, if you can link them, if you understand how this gene functions and in what pathways it plays a role, you can try to, even if you can't, let's say you have a whole chain of proteins that are uh, executing a particular function, you may not be able to change or fix whatever is broken and what is mutated in the patient, but you can do something downstream and still try to ameliorate the symptoms. That's the main hope and goal.
It's just so cool that such a system like that exists. Um, so you said that was one of your many projects. Is there any other like really cool project you'd like to share with us before we touch on the next stage?
Yeah, we did, like I said, the screen for NMJ development, for neuromuscular junction development. I really liked a lot. It was um, mostly because I like being behind a microscope and having that first glance under the scope of a mutant that no one had looked at before. At that point, we didn't even know what gene was mutated, but to just look at how, yeah, at how those patterns had changed. And you would have, like you normally had these two branches that would form almost like an X. Sometimes they had expanded to span the whole segment, sometimes they were all bundled up. And so we actually found that many of these mutants that had the scrunched up, bundled up NMJ, which, instead of nice and large, they call them boutons, which is where the neuron really forms the contact with the muscle. It's like this small circular, at least when you look at it in 2D structure. And so they would have, really, it would be like a grape almost. And we found that when mutants had that pattern, they were almost invariably mutations in a gene that was important for mitochondrial function. And so that was um, another whole realization of this screen, was that many of the genes. So one of the main sub-screens that we did was to look at genes that would cause neurodegeneration, and we would look at gene function in the fly when they were young versus when they were old. And looking mostly at, we would do that in the fly eye. So we had a specific system to look at the function of neurons in the fly eye, and we would compare young to old. And we saw that the ones that had the strongest phenotype, similarly, they would almost always map to mitochondrial genes. And so that opened up this whole new field in the lab, where we were studying how the mitochondria and mitochondrial function was important for neuronal function and to maintain neuronal health over time.
And what's an old fly?
We did this, so the we did it depended on the project, but initially it was like within the first few days of, like, you have to
Okay.
let them acclimate a little bit. That was a young fly, and then you would do it a month later.
Okay, cool.
But
So
then
it
if
shows how like quickly you can study in a fly aging, right? You're not talking
Yeah,
years like you would in a mouse. You're talking on the order of
no,
a month, and that's your old animal. Compare back to
right.
young. That's really really cool. Speaks to the
Yeah,
power
so
of the
it'd be
system.
1 to 2 months. If you would want to stretch it out all the way, then you would go to 2 months. But yeah, you can do those experiments. It still feels like a long time when you're in the middle of them, but relative compared to other, like you mentioned, the mouse studies, those are, yeah, much longer. Those are years.
Nice.
So yeah, that was uh, a whole. So yeah, I felt like during my PhD, I really had touched on many different fields, um, and um, the project I was most involved in towards the end was the muscle project that I talked to you about. Um, and so I started thinking of what's next now. And there I really was very confused with myself, with where I wanted to go at that time. I was happy with what I had done, but I didn't know what was next. I wasn't sure. I think this is a common feeling for scientists. I wasn't sure if I had what it took to start my own lab, if I would be able to open one, to run one. Um, and so I had a lot of self-doubt, and I also wasn't 100% sure anymore that that was really what I wanted still. So I started to do some self-exploration of, okay, if this is not what it is, then what is it? And I really wasn't ready to make any change just yet. So I tried to think um, through I tried to keep as many doors open as possible. And I said, okay, if I want to give myself the best chances to be able to run my lab at one point, um, then I think I need to change fields, because at that point, um, many of the papers we were sending out for review would come back with a ton of questions, but there would invariably be one that would ask you to confirm what you had seen in the mouse, where people wouldn't really believe that what you had shown was relevant unless you had also shown it in a mammal. And that, I thought, um, I wanted to get some experience working with mouse genetics. And so then I thought, maybe, the muscle is my overlap, and I'll join a skeletal biology lab and start studying how the muscle is affected in skeletal diseases in a mouse. So that's how I then joined Brandon Lee's lab as a postdoc.
And was that also in Texas or was that elsewhere?
That was in Baylor, because I
Okay.
had in the meantime also met my now husband, and so that was, I think, part of um, all of those doubts, was if I stay in Houston. I did not want to move to a different state or city at that point, because he could not, and so yeah, there was a lot of self-doubt at that point of, okay, I still call it my own sort of midlife crisis. um, and I spent a couple of years really trying to figure out what it was that I wanted. If it wasn't science, or if it was science. And if it wasn't science, then what would it be? And I think I I've talked to many people over the years. I think this is in, incredibly common. I think all scientists go through that at some point, if not at multiple points, where you sort of have to do this check with yourself of, okay, where am I? Where do I stand? Where do I want to go? And it can be really confronting and induce a lot of anxiety. I know it did for me. Um, um, but I think I came out of it stronger and with a better sense of what I enjoy doing, where I want to go, and I still don't think that I have the picture of where I will be in 5 or 10 years, but I am more okay with that, and I have a better sense of what I have to offer and how I could change direction if I would want to.
Yeah, and you
So
also
that's,
the two-body problem too, right? So it's not just
yeah,
necessarily where or if science, but then with your significant other too, of where you'll be and
yeah.
moving and where to go. Yeah, it seems like a very stressful situation. Did you start
It's and it
did you start
it's
questioning
one
it
that,
like in grad school, or was it that postdoc transition? Like,
no, it was really at the end of grad school, and so my PI told me, "Oh, you're ready to graduate," and my paper was about to be submitted, but we weren't there yet, and so I was really pushing back. And I told him, I'm not ready to graduate, my paper isn't out, and he was telling me, what are you talking about? You already have these other papers published, you don't need this one to graduate, you are ready, you should move on. And it took me some time to understand where my own feelings came from in not wanting to graduate at that moment. I did, because he told me I was supposed to graduate, so I did. But it caused me so much um, anxiety and stress, mostly because I actually didn't know what I wanted to do after. Um, and so there, um, I guess because I was on a visa,
mm-hmm.
I did not have the same choices that a citizen would have. Just for people who are unaware, when you're on a visa here in the U.S., you can't you can only work in the environment that was described in your visa. As a student, if there were intern opportunities or anything alike, those are not allowed. Um, and the same when you graduate, it is much easier to get a visa if you stay in academia. But if you would go into industry, you can do that now for a limited amount of time, which is, I think, still 3 years on what's called an OPT. But by the end of that time, you need to have figured out how to stay. And so I decided to stay in academia for my postdoc, and like I said earlier, to try and keep those doors open, and figure out what it was that I wanted. But then at the same time, really explore and try to give myself opportunities to explore different types of skills. And that's also where the ECLP came really at the right moment, because I got accepted into that program just as I was starting my postdoc. So that was a really good opportunity to actually figure out what else I was good at besides planning and running experiments in the lab.
So yeah, could you tell us a little about that of starting a new postdoc and a new lab, right? While starting a training program as well. Like, how did those two kind of come together to maybe let you the midlife crisis here of like where science, like how did that, if it did, alleviate that stress, and what would you
Yeah,
suggest for others?
it I was in a moment where I had a lot of self-doubt and really didn't think I was good at anything besides what I was doing in the lab. So I didn't see what I would have to offer outside of the lab.
Mm-hmm.
And so when um, I got into the ECLP. We were the committee that I was part of was doing community outreach and engagement, and so we um, had this monthly webinar that we were organizing, where we would give early career researchers who were part of the GSA community the chance to present their work, and helping to organize that um, kind of was really the first step of, oh, this is kind of fun, and this is something else that I've never done before. And I was part of a group of other people who were close to in the same boat, just learning new things together, being excited about it. We had a really good group of individuals together who were all um, really talented and motivated, and worked together really nicely. So it was really great to grow through that together. And then in it's a 2-year program, so in the second year we tried to think of what else we could organize. So we started experimenting with some other formats and initiatives. um, so it was, yeah, I for me it was really a great opportunity. And I'm still in touch with the people I met during those 2 years. Yeah, it's been, for me, it's really been in one of those situations or experiences that opened a lot of doors that I had no clue about ahead of time. um, and it came around the same time as when I got accepted into another program that was run by eLife, which was the Community Ambassador Program. And their program is really focused on trying to improve the research environment, the research culture. And so they accepted, I don't remember whether it was 100 or 200 people from all over the world. They were all early career scientists, and they gave them a certain amount of training at the beginning of the program, and then they basically asked the question, okay, here you have this communication platform. What do you think we could improve, looking around in your research environment, and in general in the academic research culture? What is going wrong with it? How can we improve it? What can you think of, that we need to make things better? And so you would have all these initiatives that would pop up across the spectrum. Some were looking at sustainable science, some were looking at bullying in science, some were looking at reproducibility in science. So there was a ton of different ideas. Difficult to for me at that time to kind of choose, because I thought they were all important, and I didn't really know what to pick that I thought I could contribute to. Um, but there I then sort of rolled into this initiative trying to improve the reproducibility of science. And so we developed this workshop that would teach people, regardless of which career stage they were in, different tips and tricks of how to make their work more reproducible. And that, I think that was, along with the ECLP, those experiences there really showed me that I can do different things, but also taught me how to advocate for change. If I saw that I thought something could improve, how I could do it. So together, all those things were just showed opportunity, which I didn't think at that point was there.
So the advocacy for change is really exciting, and also I would think terrifying in some ways, right? Like at least I've always heard the horror stories too of, like, if you speak up for change, you're gonna
Yeah.
anger the big wig up top. And so I guess, can you talk a little bit about that experience and how you gained your voice and weren't fearful of stepping up for what's important? Because I know there's a lot of young scientists, even in the early career program now, that come to us and be like, I see this issue, but I don't know what to do or even where to begin doing something to advocate for change.
Yeah, it's not that easy, because you have to, I think, as an early career scientist, our confidence of what we really know is very low. Even if we are really experts in different fields, we don't necessarily um, feel like we are. And we always have the idea of, oh, but who am I to say, X, Y, and Z about topic X, Y, and Z? Um, I I think what what has helped me in that sense is really that feeling of community. I was never alone, and I could always find people who felt similarly, and we would work on whether it was a blog post or an article or a workshop, we would be developing it together. So in that sense, it's sort of protected from that, big wig up top kind of scenario. But it was also at that time, when we're talking now specifically about the eLife program, that was just part of how eLife had developed their system to try and foster that trust. Um, so I felt a little bit more protected at that time, whether that was grounded, but I think what I've struggled a bit with that more. So one of the projects that I got into through that ambassador program was a blog called ECR Life, and there we it's really meant as a platform for early career scientists to advocate for change or to share their opinion, or to write how-to guides or whatever is on your mind as an early career researcher. ECR Life is a platform for that. And so we've had a lot of blog posts about quite sensitive topics, and as one of the editors, you work with the writer to try and get their message across the best way, but also in a way that is not, hopefully not going to upset the big wig up top or cause whatever social media storm, or you want to raise awareness, but in a way that you're not put in the wrong spotlight. So that has been a little bit of a, it's not a struggle, but a challenge sometimes, to figure out, how to balance that line, kind of.
So it's not anonymized, right? So your name's attached to that piece.
Yeah.
And so that's that
Yeah.
balancing act too of like how much can you say without crossing that line.
Yeah, and we have also published some anonymous blog posts, where we've said, okay, as long as an editor we know who we are working with, if they don't feel safe to publish this, but we are okay with putting it out under our name, and we say that it's an anonymous writer, we have done that too. So that is sort of a way to try and protect the writer in that sense.
Yeah, which is really important. I think jumping back to an earlier question. So you're doing your postdoc. You're also in these several programs, and now you're picking up writing as well. We talked a little bit about the beginning that you were also like for a year a science writer. We had a question at the start of how did you navigate meeting the demands of academia and science writing simultaneously?
It went actually pretty well, mostly because I was really motivated for both. So I tried to protect my time in the lab, and I felt like, what, this is my job, this is what I'm paid for. So this is, I'm not going to be writing or working on any of my advocacy projects during my time in the lab. But then my evenings and my weekends were often spent on that. It became a bit more challenging when we became a family and we had a little one. Um, that time I had to put a couple of things on pause. There were times where I think this is a very common story, but I would be doing things while she was taking her half an hour nap, or in the middle of the night, just because I was sleepless. It's motivation that will drive you in those times, and that's also what shows you what you really enjoy and what you really like, is when it doesn't feel like work. So yeah, that was a time I was just, it was also our reproducibility for everyone initiative was just funded by CCI at that point. So we had this momentum that was really picking up, and it was just exciting to be part of it all. And yeah, it was hard. I didn't really sleep all that much, um, and I've had to learn how to prioritize things and how to be more effective on certain levels, and say, look, this, I as much as I want to put my shoulders behind it, I just can't, and I will feel guilty if I I've had this several times where I'm excited, so I say yes, I'll do this, and I'll be part of that. But then over time, I see I just don't have enough time to really work on that project, and then the guilt builds up, and you get into a more negative spiral, and then that's when it's time to really think through, okay, what am I part of? Where can I really contribute? And where is it better for me to say, look, I'm sorry, but I think I'll have to step back here.
I mean, it honestly, as being a father of a newborn too, it sounds like you're being a superhero at this time, right? Like you're the postdoc, the writing, the reproducibility for science, the
Yeah,
kiddo. Like
it's I
that's a
I think
lot.
I'll stress the community factor again, because I was never alone, and it was always, we were tag teaming through different things. So with whatever time I had, I would contribute what I could, and we've all gone. I It's something that we stress with our ECR Life editorial team too. We all have moments where we have more time versus less time. And when we have less time, it has to be okay to say, look, I'm sorry, I want to do this and I've signed up to do this, but I just can't right now because, I don't know, this grant deadline came up, or whatever it is, whether it's personal, work related, or private. It doesn't matter if it doesn't work at that time. It has to be okay to have to say, I can't do it right now. Is someone else in the team able to do it?
Hmm.
And I think that's what keeps things afloat. If you can't do that, and everything fall is your responsibility, and you don't have anyone to fall back on, that's when things break, I think,
Yeah, that's
or when they become unsustainable. We'll say it that way.
especially yeah, with so much juggling so many things right at once. If it's just
Yeah.
you versus a team. So what? Just focusing a little back on your postdoc, what was the research question? Again, similar to with in graduate school, what was the thing that you're most excited or proud of?
I had a project where I was looking at how stem cells are affected. So I was looking at the muscle, but the muscle turned out to be really difficult to study, and the lab didn't have experience with it. And I spent almost 2 years trying to optimize a protocol just to look at histology, to stain, to dissect and fix and stain muscles so that they would still look intact and good, which was also the main driving force behind me getting into the reproducibility for everyone project, because this was such a seemed like such a standard technology that everyone in the muscle field was doing, but when I tried to do it on my own, it just wouldn't work. Um, so that project was taking off very slowly. Um, and then I had another project where we had a way of tracing stem cells and their progeny, and trying to see what would happen in our mouse models of osteogenesis imperfecta. And so those are mice with very brittle bones. The kids who have that disease, depending on the severity of the disease and what kind of mutation they have, they can fracture even in the uterus.
Wow.
So
Is it a
so
pervasive one? Like is it like what are the numbers? Is it more than like what you did in grad school with respect to diseases? Like how prevalent is it in the population?
Oh yeah, it's also considered a rare disease, but it is more prevalent. It's one of the most common skeletal diseases that is hereditary. It is also something that they screen all pregnant women for when they are in their second or third trimester. I forget which time. Now, just give me 1 second. Ah, okay, okay. Sorry. Um, yeah, so skeletal diseases, and we had different mouse models. This is a disease that can be caused by mutations in different kinds of genes, but they're all the building blocks of what makes up your bone. And so um, the we had generated this mouse model where we could look at the stem cells, and we had hypothesized that there would be fewer stem cells because in this particular uh, mouse model, which had a mutation in Wnt1, and Wnt regulates stem cell maintenance and homeostasis. So we thought, oh, if you mess up Wnt, then you would have fewer, but we saw the opposite. And so it was really interesting, and the project is still ongoing, so we're still trying to get to the bottom of it. But yeah, that also um, taught me the challenges of mouse genetics and how difficult it is to get different alleles in the same animal.
Yeah, it's very different than the flies, right? It's
Yeah, yeah.
I feel like that's a harder transition to go from fly to mouse than from mouse to fly.
Yeah, I don't know if I would say harder. I think they both have different challenges because there are so many different tools that are available for manipulating genes in the fly that I can imagine it would be pretty overwhelming to
Yeah.
to transition the other direction too and say, but what are all these things? I get so confused.
That's fair too. My fly bias is coming out.
Yeah. Well, I mean, yeah, it just when you transition from fly to mouse, it's a bit hard at first to say, oh, but we can do this. Ah, no, but we don't have the tools that would enable us to do that. Ah, but we can express it there. Ah, no, because the Cree line is a less selective, or so in that sense, the tools that exist for flies are more expensive. Um, you have to, at least in the skeletal fields, which is not, it has its own restrictions, or like it's when it comes to drivers that can drive expression of a gene in a very specific set of cells, it's it's harder to find them. They often are also expressed in other connective tissues, and so you have to try and figure out what you can do with the tools that you have.
And then we have a
And I,
question too in the chat of like price relation as well as to working with the mice or
yes,
flies.
yes, for sure, they are much more expensive, and like we mentioned earlier, to time wise, to work with them, you have to age them to the right time. The experiment I was doing, I was looking at how well those stem cells could differentiate in bone forming cells.
Hmm.
Um, and so we would create an injury in the skull of the mouse, and then do time lapse imaging where we would go and look a week after, 2 weeks, 4 weeks. Um, so those are very long experiments, and it's easy to lose your mouse. It's a pretty invasive surgery, so we would lose our patients in certain occasions, and those are um, frustrating and unfortunate moments, but yeah, it's
Was the imaging also difficult, or is like the difficulty at the beginning when doing the surgery, or is it each step also like pretty invasive? Just for us that who don't work with
uh
mice, like how difficult is like this kind of experiment? Because I know as science we kind of just rattle off like we do this thing and this thing, but like the logistics behind it is pretty intense, right?
yeah, it's I mean, there's a lot of training behind it too. You're not allowed to just do live surgery, they call it survival surgery, where the mouse has to survive after the surgery. You can't just get into a lab and do it. You have to get certain sets of training for it, and so that way you learn how to handle the mouse, how to anesthetize it properly so it doesn't feel pain, and then it's going to be specific. You know how to suture it back up afterwards, like the pre and the post op surgery, or procedures you get trained on, and then the surgery itself is specific for each lab or even each experiment. So there, I was lucky enough to not have to develop that aspect of it. So it had already been established in the neighboring lab, which was studying skeletal stem cells. And so they showed me everything. They were great collaborators, and they helped me at the beginning of that project too. So it is not easy, especially um, the the combination of making sure that your mouse is anesthetized enough so that it stays asleep, it doesn't feel pain, but it also doesn't wake up while you are imaging for a certain amount of time. Um, that was not easy. The actual surgery was not that hard because the skull is right under the skin. It's very accessible, and we had a small instrument to make the injury so that it would always be relatively consistent between experiments.
And it's really cool just that you are also running this reproducibility in science series while you're doing these kinds of experiments. And I think that's something that, could be easily overlooked, but I think that's just a neat thing because then you're thinking like in your experiments too, like how to make them reproducible and how
Yeah,
writing down the
yeah,
methods in a way, right, where it's followable. Because you see so many papers where, based on previous methods, and it's 6 papers back of citation, and that method doesn't
yeah.
even look like what you described,
Yes,
right?
that is definitely 2 slides actually in our workshop of trying to get people to take it. It was for me it was incredibly valuable and enriching, and it came at the right time because I was struggling with exactly those elements of doing research, and it was also the time when this platform called protocols.io had just been uh, established. Um, and so this is a platform where you as a researcher can go and write out your or and publish actually your protocol. So you can upload a protocol there. If you want to keep it private, for it is free. You have a certain amount of protocols that you could keep on your own uh, uh, profile, let's say, and you can publish them too, and then you actually get a DOI for it, and so you can get credit for that publication. And um, the big benefit is that you can really go into detail for each step and explain which reagent specifically, not just very generic now, but let's say PBS, but what vendor did it come from and what concentration was it, and go into more detail than you could in a method section of your paper.
Yeah, it's always frustrating in the publications, right, when you're cut down on the text length, and you're just, oh, I just want to write out
Yeah,
everything I did.
5 lines. And I will say that in the I guess by now 5 years since then, things have evolved a lot in the right direction, I think. So a lot of journals now let you expand your method section in supplementary materials. Um, and there are now different initiatives to try and publish your protocols for under different journals too. There used to only be Nature protocols, or there were a couple of others, but those were, like now there are many more venues for you to get credit as a scientist for the protocol you've optimized.
We have just speaking on the publication front, we have a question from the chat. How do you feel about open access journals? Do you think there's a future for them to compete with the Cell, Science, Nature, the big journals?
Yeah, that's the big question, right? That is definitely the hope. I really hope that we can get past this idea that an impact factor determines the quality of a paper. This is still stressed, I think, way too much. We have a great blog post on ECR Life about the impact factor that for me really opened my eyes, because until that time I was also still thinking along those same lines. But I really hope that we can move beyond that, um, and actually look at a paper and judge the quality of the paper itself, and not refer to its quality based on where it was published.
Yeah, that's always the hope, right? 'Cause it's
Yeah.
you default and assume. I'm guilty of it too. It's like, oh, it's in this big three, it must be good. But sometimes you dig into, and you're like, wait a minute, there's a lot missing
The
here.
Yes, I mean, there are, yeah, it's a topic we can dive deep into, but in general, there are so many ways that a journal can manipulate its impact factor. There are certain journals that publish a ton of reviews, which tend to get many more citations compared to research papers or research articles. And what people forget is that an impact factor was the way it was developed was really for librarians to figure out which journals they should prescribe to, or which that they should get a subscription to. Um, and over time, it became a substitute for how what the quality of a specific paper was. But what it really does is average out and give you a general sense of the number of citations that an average paper would get in the journal, not this specific one. And so you'll have a couple of papers that have tons of citations, and then you'll have the other outliers. But there is quite a bit of work around that, and there's been a lot of momentum gained in the last 5 or 10 years to try and develop different metrics for that. I think it's still really ongoing. There hasn't really been a good um, uh, I don't want to say substitute, but another contender that could take over. So people still are left with impact factor as the main way to look at a paper. But there are some that look at more specifically um, your paper within its own research field, because it gets also hard when you're working in somewhat of a niche field. Um, it gets harder to get your papers into those large journals.
Yeah, and if you're
And yet
then it's
cost,
the
yeah.
really the specialized journal, right? And that doesn't have the same impact factor as the big ones,
Yeah,
and
yeah,
yeah.
yeah.
So speaking of writing, um, after your postdoc, you went into science writing. Could you tell us a little
Mhm.
bit about that choice? How did the training opportunities, like at the ECLP, maybe influence that choice, and what was it like?
Yeah, it's something that I over the years during my postdoc, like I said, I was exploring in different directions to see what I would like. Um, um, and writing sort of always came back. I always enjoyed. Hugo actually gave great training and mentoring in writing. So he would really sit down next to you when you would write your paper side by side. You would come with a first draft and you would edit it together. And that was really great training in how to write and how to improve your writing. Um, and then with the ECLP, we wrote maybe sorry, maybe not as often, but there were uh, how do you say that? Like there were advertisements that we had to make for that webinar series that I had mentioned. But then I became a liaison for the uh, what is the committee called? The communication? No, not communications and
Mm-hmm.
outreach
Yep.
committee. And so there I became a bit more involved with the different efforts and the opportunities that the ECLP had for science, for people who are interested in science writing. And so here they would work together and team up within the committee to around topics that they were interested in, and write blog posts together. And so that was on one side. I got to see how that worked. Um, and on the other side, like I mentioned, I had become involved with ECR Live. So I was editing blog posts on that platform and working with writers there. Um, and so realized that that was one of my strengths, but also something that I really enjoyed doing. And so I have a friend who was or like is a science writer still, and she was part of the NESW, the National Academy of Science Writers. And she had seen a job opportunity there, and she forwarded it to me, and it was for a science writer or a science editor with a scientist. And she said, maybe you're interested in that. And I didn't know. I said, sure, I'll give it a try. I don't know what this job would entail or what, but I was a bit stuck in my projects at that time. I still didn't know what I wanted, and I by that time really thought, okay, starting my own lab, I don't think it's going to happen. I don't want, yeah. I I looked for different openings in different areas, and I was looking more into the science advocacy direction, so nonprofits, and um, yeah, there are not as many opportunities in that area. It's very difficult to find enough funding to keep those projects going over time. So you have to be lucky that you find the right one at the right time. And so I applied for the scientist. Had a really good first interaction with the hiring manager, and they gave me a writing test, which I felt really stressed about, but they were actually really nice. They gave me at that time a relatively, basically what they did was uh, they had interviewed a scientist. At that time, it was about COVID, because we were really in the middle of COVID, um, and they wanted me to write an article about it, that would integrate a couple of quotes of that scientist, and that would highlight the findings, the main findings of the paper that they had interviewed him about. Um, and so at first I was really stressed about if I would be able to do it in that amount of time, because they gave you 4 hours. But since the topic was kind of accessible and everyone would know some background about it, it worked out really well. And so I passed that test, and then they offered me the position, and I just decided to give it a try.
And so was that during the postdoc, or is that a separate position from, like, were you
That
still?
was a separate position, so I left my postdoc at that time, and that was 2021. The summer of 2021. So I had been a postdoc for about 4 years at that time, um, and I didn't see a direct. Um, I didn't see my projects being finalized into a publication anytime soon, and so I didn't see a clear path forward where I was, and I just decided, okay, let's go ahead and see what that world has to offer.
So what did the that world have to offer? What is a
I
science writer's day to day like? Like, for those interested in getting into it, what would you
mhm.
suggest? What are the potential negatives and or pitfalls, at least with your
I
experience?
Yeah, I found it actually really enriching. I felt like I got to relearn science, because I had gotten so narrowed and focused into one particular field, and now all of a sudden I had to write about immunology, about cancer, about different technologies up and coming, like um, um, new omic technologies or imaging technologies. So it was exciting. It was also hard because you don't have good writing days every day of the week. So some days were harder than others, but for um, uh, the way that the position there was structured, you didn't have to be writing every day of the week. So if you had an off day, there were plenty of other things that you could do. Like I mentioned, we had not just writing of articles or blog posts or ebooks, which was a collection of different kinds of articles. Um, but we were also making posters on particular topics, and so there you got to work with a designer to conceptualize, whether, like, I did one on the history of stem cells, so I got to talk about everything that we discovered since we cloned Dolly, which was really cool. We also had podcasts that we were developing, so I got to interview people for that. I got to learn how to edit audio recordings and how to put them together, listen to my own audio, which I found really, really difficult. um, and we also had webinars that we were putting together. So part of my job was also just to make lists of people who had recently published in a particular field on a particular topic, and then reach out to them to see if they would want to be part of our webinar.
What was your favorite part? Because it sounds like it's you've very many hats and you skills. Because And I guess the other thing too is how different of writing style is it to go from science writing papers, grants, to something like this where it's meant for a more general public.
It's very difficult, or it's very different.
Mm-hmm.
But so it's important that you have a good mentor or trainer in that sense. I had already had quite a bit of experience editing for at least a broader scientific audience with the blog post. Um, but here it was still different because now you were talking about a scientific topic rather than a research culture environment topic. So there definitely was a transition or a training time there, but I had a good mentor in that sense. Um, my favorite, it I think the post I like the posters a lot because you it was a nice combination of writing and visualizing things, and so we would make these outlines where we would say, it was kind of difficult because you had to come up with a concept of how you would put it together, and then draw or draft it in a certain way that you could then explain to a designer, a graphic designer, how you thought about it, and then they would run with it. And so that first draft, getting that back was either really good or really bad. Like, wow, this looks fantastic. This is exactly like what I imagined. Or, oh, it looks kind of what I had put on my paper, but this doesn't work. And then trying to work through different drafts to make it look appealing and interesting.
Cause it's like an
Um,
infographic, right? Like you're supposed to get a lot from
yes,
the
yeah,
visuals and like little
yeah.
text, not like necessarily giant blocks of text. And
And that was tough too because I have the tendency to want to incorporate a lot of information. So
mhm.
I always ran into that that limit of, okay, no, you should put less text here. How can I, make this or reduce this to the bare necessary or bare essential bones of what you need as a reader. And so it I think one thing that so I spent a little over a year at the scientist, and it really taught me how to think about um, your audience more than yourself when you're writing. And it's something that it's very easy to forget because you are passionate about something, you have learned a lot about a certain topic, you want to get it all out. But it's really important to think about who's going to be looking at it and why they should care. So that balance of trying to incorporate enough information to keep someone interested and engaged, but not overwhelm them, is something that takes a little bit of practice to get to.
Yeah, and we have a question in chat we'll get to in a moment. But can you talk about how then you transitioned from the writing to now your current position?
Yeah. So I really enjoyed writing, but I also felt like after a little over a year, I was ready for a new challenge. I felt like I can keep doing this, but in the position where I was, there was not really a clear path of what would be next for me, and so I was missing that. And I think that was just a combination of my own aspirations and then what was possible within the time too. Um, and so I had started to look around for jobs again, more in the science advocacy direction. Um, and this is again really fortuitous, but my previous mentor, my postdoc mentor, actually called me on the day I had a big interview for one of the positions I was interviewing for, and so he goes, "Oh, so how's it going? Do you still like your job? Are you enjoying it, or like what's going on?" And he basically had just gotten funded for this large project, where NIH, the main funding organization here in the U.S., at least for biomedical research, had asked him. This was a project that would span um, different universities. So they were trying to erect a consortium, and my PI would have one funded project of that consortium, but they had also asked him to develop a coordination center that would oversee all five projects. And so that's why he called me. He basically wanted to see if I would be interested in that position. Um, and yeah, I had to think about it because I had sort of made that break in my mind of, okay, academia, bye. And I wasn't sure if I wanted to get back into it. But I've been incredibly happy that I did, because it's been really the right mix of being involved in science, but not too involved. Like I am overseeing the experiments, and I'm helping with planning them out, but I don't have to figure out why my Western blot is not working, for example. Um, um, so I'm coordinating the project within our institution, but then I also have more leadership opportunities within the consortium. So I have to make sure that everything runs the way it should run, that we are harmonizing our experiments, and the way we do more that the way we do our experiments in the different institutions, so that we can actually gather data that we can compare and that we can integrate. And so that's been really um, both challenging but incredibly enriching too.
It almost seems like you were kind of set on that course from the very beginning, right? Working with chick embryos and flies and like harmonizing different research questions and organisms to the point where now you're just
Yeah,
like,
that's a nice way to say it. I think I had to. So I still really enjoy science writing, and I work as a or I try to work as a freelance science writer on the side now. It's really today. It's been a year since I started in the position of coordinating the consortium. Um, and it's been a lot. It's been a lot of work. It's really fun to be part of, but it is also at times very overwhelming. So I haven't been able to write as much as I wanted to. Also because those other projects like ECR Life and reproducibility for everyone, they are still active. So I'm still involved there too. And so I've sort of had to say, okay, there's only this many hours in the day. I do really try to protect the time that I have with my family. So I don't have mental brain power to work at midnight the way I did before, so I have to choose.
nice. It's a really it's a really cool story of how everything came together at the like to the current career stage. What do you think are the next steps that you'd like really like to see, like where you're working now? Like, what is the dream result? Is it like a certain paper that one works for? Like, 'cause it feels different, right, from the previous positions. And
Yeah, so what I'm working towards now is try to understand. This was like really the high level objective, right? The big goal that is hopefully not too many years, but quite a few years ahead of us, is really to understand why people with joint disease have pain. So if you have osteoarthritis, there are a lot of people who have pain in their knees, in their shoulders, in their jaws. Um, but we have very little understanding of where that pain is coming from. There are different tissues that make up your joints, and we actually don't have a good read on what's causing pain. So now we're really trying to look at where the nerves are within those different tissues, which it sounds like, oh, but you should know that already, right? Look at what we know about the connectivity and the circuits within the brain. But when you look at the skeleton, everything becomes so much more difficult to study, because you have mineralized tissues that you have to try and either make them transparent or section them. Sectioning bone is not easy. Sectioning the brain is, it's very soft. You can make very thin sections, but the bone will just splinter if you don't do something to make it softer. Um,
I'd
so
never thought of that because we had a sectioner, and you'd put an insect brain on and cuts, no problem. Yeah,
yeah,
that's
yeah, there are things you don't necessarily think about, but so the field in that sense, the skeletal biology field is not as advanced when it comes to imaging technologies, when it comes to um, different type of technologies to study how things work within the tissue. And that's a large part of the funding we've received now, is really to try and develop those technologies. And that's, I don't know, I guess I had forgotten how fun that was when I was doing that in Hugo's lab, to try and develop a technology and see how it starts to work. It's frustrating a lot of the time, but when it starts to work, it's really cool. So we're starting to get to that point where some things are starting to work, and it's just, it's again that feeling of being among the first ones to be able to look inside and see what's happening is really, yeah, a big part of what as a scientist is really motivating and just really cool.
Yeah, and developing a whole field, right? That you get to do, like, be on the forefront of that.
Yeah,
It's
yeah.
really, really cool. We have a question from the chat of what would be one question you would recommend potential postdocs, members of a lab, asking potential postdoc members of a lab, not the PI, before joining a lab, and maybe that's not just for postdocs but for graduate students as well.
I think it's incredibly important that you talk to all the members of the lab. I've experienced this when I was looking for my PhD lab myself, where I interviewed somewhere and really had a great impression. PI was amazing. The students I was talking to, the postdocs I was talking to, were all super positive. And then in passing by, this person sort of, took me aside and said, "Look, I'm not allowed to talk to you. I'm not on the schedule, but I'm a student, and I'm, leaving in a few weeks. I decided to quit, and this is the list of people who have started their PhDs in the lab, and this is the list of people who have completed their PhDs." And I wasn't aware of that. But there are a couple of things that person told me at the time where it became very clear to me that maybe I get lucky and I become the PI's favorite, but the chances are very real that this is not a good environment and that I shouldn't come here. So I think that is even more important than the right question to ask. I think you want to ask about the environment, how the PI maybe gives you feedback. Um, um, what else? Uh, I think every lab is going to be so different. What kind of what the expectations are of the PI towards you, how rigid those are, and how flexible they are. Those are things to ask about and to be mindful of, I think.
Is that a similar vein to the science writing as well, of like knowing that the scientist is a
Yeah.
good
Yes.
environment?
Yeah. Knowing, making sure you talk to everyone. Yeah, I think that is just, it's not as easy to figure out who is part of the team
Mhm.
in every environment. Some labs have websites where they list everybody. Not everyone has that. The same is true for companies and for different groups. There, it tends to be even harder to figure out who is part of a specific group. But when you're interviewing, you can ask your different members also. So who else is part of the team or whatever, and so that way you can try to poke holes into, um, you know, if someone is trying to hide things, you can try to figure that out.
It feels like a game of cat and mouse detectivizing that you shouldn't have to detect at that stage,
Yes,
right?
yes, but unfortunately, that is reality. There are people who, I mean, there are a lot of, when you are coming out of your PhD or your postdoc, there are a lot of people who are really ready to leave, and they don't think they have, a good CV, or they don't have the right skills, and so please take me. Um, it's a bit risky, because there are also organizations who try to take advantage of that and who underpay you, and who will just get you in and then put you in a position where it's not as beneficial to you as you thought ahead of time. You can't always know that before you start. You can try to ask as many questions as you want, but that is something that you'll have to take the plunge at some point. I think making sure you talk to everyone is one of the best ways to try and try to find maybe previous lab members, or people who worked at that company before and try to figure out what experience they had. But I think the exit rate is an important one to try and figure out.
Yeah, that's a really good call. I'd never thought about like the ratio as well. That person who pulled you aside was like, here's how many started, here's how many have actually finished, here's the number that left. Like that's not a
Yep,
metric
yeah.
that's tends to be advertised, right?
No, that never gets talked about. Because it's also, I mean, a lot of things happen, right? Whether that's the PI or the, there sometimes it's just not a good fit. But if there's a strong trend, it should raise a flag that you should try to investigate.
Yeah, yeah, that's a very sound piece of advice. As we're rounding out, if anyone has any other questions, please feel free to drop them in the chat now. Nele, I just wanted to ask you, what is like one big parting thought that you'd like to leave us with? Like you've had an amazing career journey thus far, from so many different lab environments, model organisms, questions of study, even fields. Like, what is your big takeaway for folks?
Um, I I guess don't be too negative about yourself and too pessimistic about your skills. It's very difficult. It's very easy to do, but you do have many more skills than you realize. Um, and the second thing I think would be to try and engage with your scientific societies. They very often have a lot of opportunities for early career scientists that can really help you in ways that you didn't expect or anticipate. Whether it's to build your network or to build some skills, or just for mental support when you need it. I've really made a lot of friends through the different organizations that I've teamed up with, and that's really gotten me through the more difficult times.
Yeah, like how you were saying that it's the support network that was behind you for a lot of these
Yeah.
steps. And it's something that I think, at least in some instances, feels very isolating, where there's not always a team behind you. And it's nice to hear that there are the professional societies that you can follow up with, and they can be supportive enough of a network that you don't have to tackle everything alone.
Yeah, and they don't necessarily have to be right in the lab with you to understand what you're going through. So I think there are also a lot of resources online to help you with different steps, whether it's getting through if you're going through an academic midlife crisis like me, or you're just trying to explore or build skills or intern somewhere. Um, there are many more opportunities now to help early career scientists with that. That field is really developing nicely.
Nice, that's it. Awesome. Well, Nele, thank you so very much for taking the time to speak with us today. I
Yeah,
know.
thank you for inviting me. It was really fun. So mostly I'm active on LinkedIn still. So I think that's the better platform. If you want to reach out, you can find me there, and I'm happy if you have any follow-up questions or anything to talk to you.
Awesome. Thank you so very much.